Hello, everyone, and welcome to the first round of talks for the day one for the general track. I'm Ash, I'll be session chairing this block, and for our first talk we have Nick Moore talking about the Primordial code. So everybody give a big warm welcome to Nick.
Thank you. Okay, just to start off, content warnings. We mention infectious and genetic diseases, including cancer and COVID, but not like any gruesome detail or anything like that. Just content warnings. All right, my name's Nick Moore. I'm a consultant. I work in software development and systems architecture for a number of clients in a number of industries through my own company, Nemote. I've been doing that for a lot of years now, and it's awesome fun. You should try it. I've worked I've presented here previously about MicroPython, about the Internet of Things, about visual programming languages, about functional programming in JavaScript, not at this conference, at LinuxConf, and about how to do NoSQL in an SQL database. And now I'm having the luck of being able to try my hand at being a bioinformatician. To start with, thanks and apologies to all my colleagues at Walter and Eliza Hall Institute of Medical Research and the University of Washington Genome Sciences and the Brockman Batty Institute. The mistakes and oversimplifications in this presentation are all mine. They've been incredibly patient, helping me learn and find my feet in this really amazing field. There is, or at least there will be, the slides, the notes and the errata on this presentation up at that URL on that QR code. So it'll be updated soon enough. And that way, any really gratuitous mistakes I make, I can at least apologise for later. All right, let's talk about bioinformatics. What is bioinformatics? Does anyone other than Alan and Estelle know what bioinformatics is? Cool. That was way more people than I expected. Hopefully you don't all work for WeHi. So bioinformatics is the study of biological systems kind of numerically, and because biological systems are a smidge complicated, that basically involves computers. We're really lucky computers and biology have kind of developed in parallel over the last couple of centuries. So it dates back to sort of the invention of computing in the 19th century, and the idea of data as this continuous tape for telegraphy and things like that. And at roughly the same time, we were discovering the idea that inheritance of characteristics in plants and animals was governed by sort of discrete inheritable units. They weren't really labelled genes yet, but that's what we're talking about. We're talking about genes. And Darwin was working on natural selection and how all of this could actually end up with the diversity of life we see. At the same time, we actually did discover the DNA molecule and discovered there was this enormously long, wiggly molecule, and it wasn't very interesting, and it just looks like boogers, but we had no idea what it was for. I just think it's funny that at the start of this story, we have DNA already lying around and everyone goes, oh, gross. Anyway, at some point or another, though, people worked out that these inheritable units had a relationship, ones that were more likely to be inherited together, ones that were less likely to be inherited together. And with a paper and pen, people worked out that there was like a linear pattern to this. These things there were closer ones and further ones, and the distances kind of added up in a long line, which is really interesting when you think about it. Koltsov proposed that this was a giant hereditary molecule, a molecule that was passed down from a parent organism to a child organism, and that must be how heredity works. I don't think he knew it was DNA at that point. It's one of those things where the Soviet world and the Western world maybe weren't talking that much. Unfortunately, that ran afoul of Lysenkoist, Lamarckist theories of evolution, and so he didn't go too well. At the same time, Alan Turing was working on the fundamentals of computing, and he came up with the idea of a universal computing machine that went back and forth along a tape. It's not really DNA, but it's an idea of this linear organisation of information that was being developed at the same time. Boolean algebra kind of got developed, and the theories behind that by people like Claude Shannon. And then, of course, the war distracted people somewhat into cryptanalysis, which ended up giving us general purpose computing, which was kind of awesome. In the 40s, Frederick Sanger, I think working on a theory that proteins were what you inherited, worked out how to sequence proteins, which was pretty awesome, and he won a Nobel Prize for it. But it turned out that that's not actually how things are inherited. Oh yeah, and in the meantime, Alan, after the war, goes on to work on morphogenesis, which is bioinformatics. So obviously this is an interesting field that's developing. In about 44, DNA turned out to be the carrier of genetic information. That horrible booger-looking molecule turned out to be actually where all of our heredity comes from. And Watson, Crick, and Rosalind Franklin discovered that structure, managed to derive a structure of the double helix that you're familiar with and you'll see in a minute. Frederick Sanger, not to be put off by the fact that the molecule he's working on wasn't the source of heredity, goes and works how to sequence DNA, wins another Nobel Prize, you know, just another Tuesday. And then in the 2000s, we developed DNA editing. We worked out mechanisms that allow people to actually edit DNA molecules, change genes. That's pretty awesome. In recent decades, we've worked out how to actually change genes in a living cell, how to change genes in a living person, and this has actually been used to cure people's genetic diseases, which is, I think, just amazingly awesome. All right, so to make any sense out of this presentation, we're going to have to teach you a bit of biology, about 4 billion years of biology in about 10 minutes. So hang on. Organisms are made of cells. A cell is like a little tiny computer. It's an encapsulation. It has a bunch of code inside it, which we call genes. That code makes proteins, the little machines that make the cell work. The cell is sort of defined by its membrane, by its edge, its outside container that keeps the outsides out and the insides in. You can think of that as a bit like a firewall. It has various ports through it called channels that let information in the form of molecules come in and out of the cell under the cell's control. So the cell kind of controls its own gateways and things like that. And that's really what a cell is. It's a collection of stuff in one place. There are different kinds of cell. That's a bacterial sort of cell, typical bacterial cell, typical animal cell, typical plant cell. They have a lot of things different, but they have a lot of things in common, which is largely this idea that the genetic information is hoarded very carefully in the centre. All right, DNA itself, the molecule we're talking about, is indeed a very long molecule. This is just a little section here of a very long spirally molecule, meters in length, you know, that is composed of these sort of bases, these subunits, and the subunits pair up. So it's like a very long tape. Instead of 2 bits, 1 and 0, we have 4 bits, A, C, G, and T. And they complement each other and build themselves up like a ladder. So you get that idea of that symmetry. That's more or less exactly what Koltsov described, funnily enough, even though he had none of the equipment required to see or work out any of this stuff. He went, oh, you know, if we're going to record all this stuff as some very long molecule, it'll have to zip together with some other very long molecule to stabilize. Anyway, that's pretty amazing. When I talk about the genomes coming up, I will talk about it in terms of base pairs or mega base pairs or whatever. A base pair is effectively 2 bits because there's 4 options for base pairs, so therefore it's worth 2 bits of information. So that's just to give you an idea of what the currency here is. So in about 77, we started being able to sequence whole organisms. The first one sequenced, I believe, was a bacteriophage, a virus that attacks bacteria. It has only about 5 kilobases. It's single-stranded RNA, I think. And only 11 genes. Anyone here who is a programmer of very old computers will sympathise. Those 11 genes actually kind of overlap each other and they interfere with each other. And it's very compact because it manages to fit enough potential into such a tiny number of instructions. It's incredible. A couple of decades later, we're sequencing much more complicated organisms like E. coli, so pretty common human gut bacteria, which have millions of bases, and yeast, ask me about my yeasts, which has millions of bases and thousands of genes. We eventually work up to the fruit fly, which is a an organism beloved of biologists because you can catch them in a milk bottle and breed them and mail them to people and things like that. They're very good to work with. They have hundreds of millions of base pairs, and eventually, in about 2003, we worked up to sequencing a whole human genome, which is like 3 billion base pairs. As a bonus, we all have 2 copies of it we're carrying around, that forms about 20,000 genes. So a human genome is quite a large project. This is just a chart showing a kind of an idea of the different species, and part of the point of this chart is actually the kind of the opposite of what I just said. Mammals are down here in this little region here, but here's flowering plants, which actually turn out to be like way more complicated sometimes, or not. You know, there's a huge variation in the size of genomes between species, I think is the point of that one. But the funny thing about it is we're all using basically the same genetic code. There's so much in common between different organisms. This is, I've been told, a compulsory slide that must be included in every single bioinformatics presentation. This is the cost to sequence a human genome. It falls precipitously, but wait. Those of you who aren't terrible science nerds may not have noticed that that is a log scale. The top point there is $100 million to sequence a single human genome. And it falls by this end to less than $1,000 to sequence a single human genome. That's kind of awesome. So, I mean, that kind of underrepresents. If I plotted it on a linear scale, it would just fall like a log and go across there. This is really exciting for a couple of reasons, one of which is, of course, it allows us to do precision medicine, to really analyse one particular patient and really get to the heart of what the hell is wrong with them, and maybe find a specific cure for that specific person, and that's really cool. But also it enables us to look more broadly at what a human genome is. The Human Genome Project has like one sequence from, it's combined from a couple of people, I think, but it is basically representing a person, right? The pangenome is like a representation of what is all of the possibilities for healthy humans worldwide, and it opens up the possibility of doing medicine for people in developing countries, for example, who are not really in the market for precision medicine, but maybe we can at least understand conditions that are more applicable to them more easily, which I think is really awesome too. And so these pangenome projects are rolling out to look at thousands of people, tens of thousands of people, hundreds of thousands of people across the world's population, and to build up a picture of what all of that combined might be, all right? Okay, so let's talk about genes, let's talk about evolution. The reason we can evolve is because we can change, and the reason we can change is because we can make mistakes. Mistakes creep in whenever a cell replicates, whenever a cell receives like radiation damage or something like that. Viruses can write themselves back into your DNA. Most of these changes are very unhelpful and not good for you, but in evolutionary timescales, the good outweighs the bad, and you end up evolving new abilities, new whatever. Not quite like X-Men or whatever, you know. It doesn't help an individual very often very much, but over billions of years, you get somewhere. So the kind of changes we're talking about are like substitution of a single base for another base, insertion of some bases, deletion of some bases, or just duplication. Sometimes the mechanism gets a bit gummed up and you end up with little duplicate runs. So as a programmer, you can kind of imagine doing this to your own code, right? It would very often not be helpful. Every now and then, it might turn out that it actually improves something. The duplication's especially interesting. If you copied a function out of a piece of code, and then now you have 2 copies and they both kind of are doing the job, then that frees you up to have one of them change a bit more without getting rid of the job the first one was doing. So that's part of the mechanism of evolution. I think it's really interesting that if replication and radiation protection and all that stuff was perfect, nothing would ever evolve. We wouldn't be here. The system has to be imperfect for it to work. I think that's amazing. Bacteria have an even better trick up their sleeve. They do horizontal gene transfer. A bacteria can basically build a thing called a plasmid and inject it across into one of its neighbours using this sort of little hairy tube things and give away some of its genes to its neighbours, not even necessarily the same species. This seems really remarkable because it means that bacteria are the first sort of free software advocates. What's really interesting about this one, right, is that it's not the bacteria's survival that matters in the long run, it's the gene's survival. If I'm a bacteria and I have a gene for antibiotic resistance, and I give that to a neighbour, that neighbour can develop that antibiotic resistance, that gene will now be replicated by that neighbour. Even if I now get outcompeted, the gene lives on. And we've all seen open source libraries that do this too, you know. So I want to talk about this concept at the centre of biology. It's called the central dogma of biology. The idea is that DNA gets transcribed into RNA, that gets translated into proteins, right? It's very orderly, very neat. DNA turns into RNA, turns into proteins. You know, you have code, you compile it, you link it, you run it. Fine. Of course, there's also retrotranscription. That's when a virus writes from RNA back into DNA. There's also replication, where DNA gets copied into more DNA, so it's not okay. And also sometimes RNA actually has a useful job to do itself. It's called non-coding RNA. So sometimes you're using think of it as a data segment in your code. It gets used directly rather than being turned into a protein, but that's fine. That's not too complicated. Except for RNA polymerase, right? RNA polymerase is a protein that does the transcription, does the job of transcription. So I think that's okay though, because we've got the protein at the end, the thing we just compile and run, and that's our compiler. Hang on. And then also RNA needs to be spliced, and splicing is kind of complicated, and it's done by proteins. And also when you do the translation, that's done by proteins and non-coding RNA. So we need to run the compiler in order to run the compiler. This starts to get a bit tricky. The only reason this works at all is because it's bootstrapped. The cell that you grew from, the oocyte, is full of these logical units, enough to keep building itself for long enough to get itself up and running. It's kind of a little bit like a boot disk. There's enough of an operating system on there that it can make another boot disk and another boot disk. And copy errors kind of creep in. So we talk about this as bootstrapping from the metaphor of pulling yourself up by your own bootstraps, which is of course impossible, but it raises the question, when did the first cell manage to do this? And this is a thing we just don't know. There's no source control in biology. We can't check out the first version and find out. It's very frustrating, but we can look at the common features. Oh, that's not the right slide. We can look at the common features. This is actually one of the common features of all these different branches of life, and so what have they all got in common? And we can kind of theorize this existence of a common ancestor that must have arisen at some point. Possibly it had RNA instead of DNA and sort of started halfway through. Possibly a lot of things. And that's where all life kind of evolved from in its different ways. This is just a little example. I was talking before, I said, proteins are like little machines, I said before. And I just wanted to demonstrate to you that this is not a metaphor. This is not one of those whole endofunctors in the whatever. This is an actual real machine. This is a thing called ATP synthase. It's a protein complex. The different coloured bits are different proteins. They all lock together. They build a little machine. It has a rotating crankshaft, and it literally pumps, either takes protons, push it around and it makes ATP, or ATP pushes around and it pumps protons. Either. It's like an engine in every single cell of every single organism, and there's slightly different versions of it around. Again, not great source control, but it is one of the basic kind of units of what we call life. You could also say that life is just ATP synthase's way of making more ATP synthase. Depends on which perspective you look at this. Okay, translation. I talked about translation before. Translation happens inside when you turn RNA into a protein. There's like a lookup table of what bits of RNA turn into what amino acids, and then the amino acids get assembled into a protein, into an exciting little machine. This looks a little bit computery, doesn't it? It's like we've got different encodings. We've got start codons, which say where to start running. We've got stop codons that say when to stop running. And it kind of looks kind of computery, a little bit like a decode table from an early microprocessor or something like that. But there's no loops, there's no branching, there's no anything. So how the hell is this a program? You can't really have a program without loops and branching and all of that sort of stuff, can you? the answer is that the chromosome, the thing that connects all the genes up, isn't just the genes. It's also control regions, like a library file. There are sections that say how much of this gene you should, or how much of this protein you should make using this gene. How much should this gene be activated? How much should it be repressed, etc., etc. And also how should it be spliced up? And all of that can vary based on the action of either external stimuli or other things happening inside the cell, other genes expression and so on and so forth. So it's actually really complicated. The program is in the interactions between genes. Just as a super simplest possible example, in E. coli, there's a group of genes that make enzymes that break lactose down. Lactose is a sugar, but it's slightly more complicated sugar. E. coli would really rather just eat glucose lollies rather than drink milk. But in the presence of lactose and the absence of glucose, it can make an enzyme that breaks the lactose down into glucose and galactose. But anyway, but there's no point in wasting its energy making those enzymes if they're just going to lie around the place being useless. So it has this little bit of logic in its genetics that says, okay, if there's lactose hanging around and I don't have enough glucose, then make some enzymes to convert them. So it looks a bit like that, except being biology, it's all squishy. None of it is just logically Boolean or anything like that. It's all about it enhances it a bit, it represses it a bit. It's all very variable and squishy. That's the way of these things. And this is like the simplest possible gene regulation. Most gene regulation maps looks more like something like that, where you have very many genes enhancing or suppressing other genes, many outside influences coming in, many causes, many effects. This is not happy programming territory. This is real spaghetti code stuff. And even picking apart the simplest one of these relationships is extremely difficult and extremely complicated. So, I mean, I think this is one of the frontiers of biology at this point, is understanding how these things interact and building these maps and trying to get an idea of what the hell is going on inside the cell. All right, one last little example about biology sort of existing things. So I talked before about all these different forms of life depending from one place. A really interesting thing happened about 2 billion years ago, I think. 2 billion years ago, where some particularly kind of entrepreneurial archaea absorbed some other aerobic bacteria and just put them to work. You know, they set up the first companies. They were the founders. So they moved in, the other ones moved in, they got protection in return for basically providing a lot of energy to the cell that contained them. And you have this encapsulation going on. So inside a eukaryote, which is, I should just explain, eukaryotes includes animals and plants and yeasts and slime molds and all sorts of things, and humans, so that's nice. Inside the eukaryote cell, we actually have a lot of encapsulation going on. We have a nucleus, which is like the central kind of most defended part of the cell. That's where the DNA lives. That's your kernel, the code. That's the most protected part. Around that, you have other like user space parts of the code in the cytoplasm. And you also have these mitochondria, which are running separately out there, doing their jobs, a little bit like a container image or something like that runs separately. Weirdly, they're running an older version of the operating system. If you think about all of this translation table stuff and all of that as an operating system for a cell, the mitochondria are still running the one they came with, or some version of it. It uses a slightly different encoding. It has its own enzymes, its own everything inside that little encapsulation. They even replicate themselves. As the cell replicates, they also replicate and send some copies this way and some copies that way. So they're in there and they're replicating along with the cell, but within their own thing. Plants do even better. They did it twice. They also absorbed a cyanobacteria, and that's how they do photosynthesis. So plants are actually way ahead of us. I believe there's some algae that's done it 3 times and also has things called nitroplasts or something, which are basically another thing. Oh, wow, this is good. Many, not all eukaryotes are multicellular. Specialization is done at runtime. We all have the same genome in every single cell in your body. At runtime, at the time you are alive, your cells will work out what they're meant to be doing. They will shift into that mode. They will do that job. They communicate with each other, exchanging messages through those cell walls. If a cell is like a computer, then your body is like a whole network of computers interacting all the time. Even funnier, things like slime molds and some bacteria basically can form big groups when times are tough, and then they can disperse into individual cells when they'd rather do that. They're very interesting that multicellular life turns out to be kind of, optional. All right, applications. Biology is really cool, but it's awesome that we can actually do cool things with it. For example, we can look at genetic disorders, right? So there are various genes. If that gene doesn't work as well as it should, you may have various problems with that function in your body. So for example, G6PD is an antioxidant. It makes a protein called G6PD, very imaginative, thank you biologists, which is an antioxidant. If it's faulty, you might get a thing called hemolytic anemia, which is where your red blood cells explode. Extremely metal, quite painful, I imagine. Not a very pleasant thing to have. You probably don't know if you have it unless you eat the wrong thing and wake up in hospital, I imagine. What we want to do is we want to find out about those variants before we have people lying around in the hospital. So the kind of experiments that I've been working on with people are taking genes like G6PD and altering them. You take every possible base in that thing and make every single one-letter typo that could possibly happen in G6PD, right? We run through those one-letter typos and we build a library of those one-letter typos. Then, ask me about my yeasts, we insert them into yeasts. This is an awesome thing. A yeast, a single cellular little thing you make beer with. This is brewer's yeast. Can run the software that runs a human just fine. They're on the same operating system. I think that is just stunning. The nice thing about yeasts is they double in population every hour and a half in ideal conditions, and you can kill 1 billion of them before lunchtime and no one cares. This is awesome for science because, this is what we like to do. This is the experimental setup. It's really just a fancy homebrewing setup called a turbidostat that keeps the turbidity, the number of the population, the same. The yeasts think they're growing indefinitely and infinitely. We feed them, then we torture them with bleach and we see which ones survive and which ones don't. And from that, we can work out which of those variants we introduced really jam up the works and which ones aren't that relevant, which ones don't really matter. We can compare them by looking at their population over time, and we can see that some things really just fade out, some things thrive. Some things are in the middle, they're kind of mediocre variants, which is even more interesting, I think. And what we can do from that, we can build up a map of those variants. And just to zoom in on that bottom graph there, this is the map showing the blue ones and ones we were pretty sure were going to be duds. The red ones were ones we were pretty sure were going to be fine. The grey ones are like all of the population we tested. So we've been able to sort the population of never before seen variants into good and bad and kind of mediocre, which is really interesting, and it has really good clinical meaning for people. Is this a thing I have to worry about? Is this not a thing I have to worry about? Other genes this has been done for are things like tumour suppressor genes and stuff like that. So a good or a less effective gene might deeply affect your odds of getting cancer in life, which is a thing that is good to know. All right, and we can build up a map of what part of the protein looks like it does what, which was what that is. If that looks incomprehensible to you, welcome to the club. I'm the numbers guy, not the squiggly drawings guy, so, All right, I'm going to rush through to one more thing. I think I'm just going to run through the question time, sorry. Ask me later. We want to be able to debug these things. It's a really complicated system, but we want to know how it works, right? Who's done hello world debugging? Print statement debugging? Yeah, it's awesome, isn't it? Sometimes you just have to. If you've ever worked in embedded systems, though, you've discovered that bugs happen so quick that the first H doesn't even have time to come out of the UART before the whole thing crashes and restarts. So what we do there is we light up LEDs instead, right? LEDs light up one after another. Hell does this have to do with cellular biology? Well, jellyfish. Jellyfish fluoresce. If you take a fluorescent jellyfish protein and you couple it to the gene you care about, then when that gene is expressed, that protein is also expressed and it glows. And now you can sort the cells by how much the protein was expressed, how much the gene was expressed, sorry. You can sort them and count them that way rather than just counting their population going up and down. This is a real experiment, it's a technique called VAMP-seq, and you can use this to analyze what's going on in these cells. All right, this is another cool example. Who's got an email that looks a bit like this? Not one of those characters is ASCII. They're all random, weird things from different languages. They get past your spam filter because none of the words you were looking for are in there. You can do the same thing with DNA or with RNA. In messenger RNA, which you're all familiar with, I imagine by now, it's been on the news, it's very scary. It's not very scary. It's in all your cells. It's the instructions for how to make a protein. You can, normally your body would filter that out. It would not accept that mRNA, but it's been discovered that if we just replace some of the bases with bases that look a lot like that but aren't quite the same, it gets past the spam filter, but the cell will still read it and it will still go and make you COVID spike proteins that you can then have an immune response to. This technique was pioneered and discovered largely by Catalin Carrico, who in the 80s was working on dual-stranded RNA, moved to the US under somewhat dubious visa circumstances, went on to research mRNA, went on to work on RNA pharmaceuticals. I'm hurrying this up because we're getting close to the end, including the pseudouridine technique, which was invented in 2005. And then went on to be the mRNA vaccines we all know and many of us have experienced. And she won a Nobel Prize in Physiology and Medicine for that. This is my thank you slide for that. It's made my life a lot more pleasant, and I'm sure a lot of other people too. mRNA vaccines are now being looked at for many other purposes. Oh yeah, I ruined my own joke. Many other purposes, including cancer therapies and things like that. Since I've been talking about biology in this software conference, I want to move on and talk about politics. I mean, not really, but sort of, right? All of this stuff has only been possible because of open collaboration. This is something that should be very dear to our hearts in the programming world as well. People send each other samples, people send each other fruit flies, people send each other sequencing data, people share and collaborate. And this stuff is not possible while hiding in your basement, not working with other people. The concept of this immunotherapy for cancer is incredibly important. A lot of these therapies are easier and cheaper as well than radiotherapies and stuff like that. So there's huge potential to rid the developing world of diseases like malaria that are horrible and terrible and all of that stuff. And really the applications of this stuff are bounded only by our imagination and our willingness to just have a go at stuff. Spend a couple of decades working on something that nobody else thinks is going to work. You might end up curing a pandemic. You never know. All right, thank you very much. I think I've run through all my question time, possibly precisely, possibly not. So thank you very much, and please ask me questions after the end of this session. Thank you.